Hi, this is Unit 1 in MLT 240, which is the second portion of micro. I'm going to cover the only the first half of Unit 1, and I think this goes through page 15 of your lecture notes. So the first page, there's some terms. Parasitism, this is a relationship between two organisms. One is the host. The host could be injured or not, and the other is the parasite. So with the mosquito is the parasite, humans are the host. An obligate parasite has to have a host to live. A facultative parasite does not have to have a host. They may live in a host, but they can exist free living in water or in the environment. An incidental host is when a host is infected and they're not the normal host. A definitive host is where the parasite has the adult and sexual reproductive phases. They can multiply in the host. An intermediate host is where the larval or asexual reproduction occurs. A carrier is a parasite, is when a parasite is in the host or on the host and there's no signs of infection. Vectors are usually arthropods like ticks and mosquitoes that can carry some other type of parasite or bacteria or other organisms that can be transferred to a host. An ectoparasite is found on the outside of the body, and endo is found on the inside, like if you were to swallow it and it gets into your intestines. The Filariform larva looks like a small, tiny worm. You probably are not going to be able to see most of these by the naked eye. You have to have a microscope. But these are the infective, non-feeding stage. These last three terms are the major phases or the major stages of most of the parasites. There are a couple that don't have a cyst stage, and then there's some that don't have a Filariform larva stage. But these are the three stages that they can have. This is the Filariform larva. The two stages that most parasites have are cysts and trophs. The cyst is usually a round or oval shaped organism. The troph is a little bit more elongated and it's shaped differently. They usually have pseudopods that stick out. They're not going to be all pretty and round all the time like the cyst. Cysts are usually found in formed stool, and it indicates an inactive or a carrier state. The troph is usually found in watery stool, and this is the active feeding form of the parasite, and it's associated with an active infection. Now, even though you can see the cyst in formed and the trophs in watery stools, it's possible to see them in any type of stool. It just depends on the patient condition. Okay, specimens are usually stool. We do get urine and other types, but the majority of our specimens are stool. The stool needs to be free of anything besides stool. It cannot have urine in it. It can't have mineral oil. Barium sulfate comes from when the patient has been to x-ray. Antibiotics will harm the parasites so they won't look exactly right. The stools for wet mounts need to be examined within 30 minutes. if they cannot be examined, they have to be placed in these transport vials. The 10% formalin is used for wet mounts. That's the pink one. The PVA is usually what people use. That stands for polyvinyl alcohol, and that's this one. This one is for culture and sensitivity because they're usually ordered together. And these come in different colors too. This one is pink and blue, but the ones I have are pink and gray. The formalin is for wet mounts. The PVA is for stains. The SAF is also for the stain, and that is sodium acetate. But like I said, PVA is usually used. Parasites in specimens other than stool, you need to know which one of these organisms comes from the type of specimen. Usually when we look for pinworms in kids, we do Scotch tape or cellophane tape procedure, and this has to be that clear tape. It can't be there are some, I don't know if you've noticed, but there are cloudy tapes that are in the stores to purchase, but we need the clear one so we can see the organism good. But this is for the pinworm, the Entamoeba vermicularis, and I think we went over this, but how you do this is usually the parent does it, and it's supposed to be done before the child wakes up first thing in the morning, and they stick the Scotch tape to the outside of the child's anus, and then the tape is put on a slide, and we look for pinworm eggs. The organisms or parasites that we look for in urine are Schistosoma hematobium, Anaerobius vermicularis, and Trichomonas vaginalis. You should remember these from urinalysis. Sputum, Strongyloides stercoralis can be found in sputum. This organism can travel to the lungs, or the patient coughs it up, and when it travels to the lung, the patient can cough it up and then swallow it back down, so it can be seen in stool or sputum, but this one infects the lungs. Sigmoidoscope is good for amoebas and cryptosporidium. Duodenal aspirates, you can see Giardia, Strongyloides, Fasciola, Cryptosporidium, and Isospora. Whenever we first get a specimen, one of the first things that we have to do and we have to document is what it looks like. So we always will document if it's soft or liquid, or if it's formed, what the color is. So with the consistency, you either say that it's soft or liquid or formed. The soft or liquid tends to have the motile trophs, and it must be examined within 30 minutes. The formed usually has the cyst in it, if anything, but it could have the trophs in it too. You need to examine it within 2 to 3 hours of passing, and it needs to be refrigerated if you're not going to put it in those transport vials. The color, if the stool is really black, this could indicate an upper GI bleed, and that's from old blood. If the blood is red, it could indicate a lower GI bleed. If there are if there's parts of the specimen that look bloody or mucus, then these are the best parts of the specimen to use. The specimen should be put in formalin for concentration in both the PVA and the formalin vials. With the microscopic exam, A lot of hospitals will do a direct wet mount. This has to be done within 30 minutes of getting the specimen, and then you take a small amount of the specimen and you can mix it with saline or iodine, and there's two types of iodine, Lugol's or Dentonus iodine, and you look for motile trophs in these. This is the best way to see motility, is from a fresh specimen on a direct wet mount, and you just cover slip it and look at the specimen. Okay, concentrated methods. Since parasites can be rather rare in the specimen, there are some concentration procedures that can be done so that we can see them if they're there. So all of these are based on specific gravity. They're good for cysts, eggs, and larvae, and the types are the zinc sulfate, formalin ethyl acetate, and the Shether sugar flotation. The zinc sulfate, it's not good for percolated eggs, and you'll find out what that is in just a little while. Or schistosoma eggs. The parasites will float to the top because it's based on specific gravity. The parasites float to the top. In the formalin ethyl acetate, the parasites settle to the bottom of the tube, and this is an example of the ethyl acetate procedure. You add formalin and ethyl acetate, you shake it and you centrifuge, and these are the layers that you get. The ethyl acetate will be at the bottom. Then there's a bunch of debris, which might be stool or food, just junk. Then there's a layer of formalin, and this is the sediment that you look for the parasites in. This is the procedure that a lot of hospitals use out of these three. The Shether sugar is for Cryptosporidium. After you do these procedures, for this one you would take the top part off and look at the sediment at the bottom. So you would take a glass slide and put a drop on the slide, and you want to use 2 drops. One drop you cover slip and you look at it as it is. The other drop you add a drop of iodine to go with it. Some of the parasites are easier to see if they have iodine in them. So you cover slip them and you look at both of them on low power first, and you scan the outer edges because the parasites will tend to go to the outer edge first. Then you look at the entire slide and you switch to high power when you think you've seen something suspicious. Okay, and again you're looking for, most of the time you're looking for trophs, and you're looking for cysts. The trophs are motile. After you have used the formalin and the ethyl acetate, you may not see the motility. The motility is best seen when you do the direct wet mount from the specimen. But these are examples of what trophs look like. They're a little bit more elongated. This is a pseudopod here that helps it to move. This is another one with a real long pseudopod down here at the bottom. This is the cyst stage, and these are more round. They may be oval. They're a little bit more refractile. They're 2 to 4 times the size of the red blood cell. The cysts are better seen with iodine, so you want to look really good for those on the iodine. These are the permanent stains that you would use from the PVA vial of the stool. These are permanently stained smears. They last a long time, so you could go back to them if you needed to. The iron hematoxylin stain is a type. The most common, and a lot of the pictures in your book and the pictures that I use, are trichrome stains. It's the most common. They stain good with this, but the other ones are used. The Gamori silver stain, Giemsa, and Kenyan modified acid fast stain. This is the AFB stain, and it's used for Cryptosporidium and Isospora, and they stain hot pink just like the Mycobacteria do on those slides. But it's not used for any other parasites besides Cryptosporidium and Isospora. You don't need to know this procedure, but I just wanted you to see how long it is. It's a lot longer than a gram stain, but this is the steps for the trichrome stain. Notice that you leave it in the mordant for 30 minutes. It takes a long time to do these. In the trichrome, the cytoplasm of the cyst and troph stain blue-green. The nucleus and red cells are dark red to purple. The ova and larva, the ova are the cyst, and the larvae are the little worms. Those stain red, or they may look pinkish. The background debris, bacteria, and yeast stain blue-green. And this is a trichrome stain, and these are both trichrome stains. This is the blue-green that it was referring to. That's what color they are. Okay, if you've noticed when you're looking in the microscopes, there is a little ruler in some of the eyepieces. They're not in all of them, but if you have a little ruler, this is called a micrometer. And the micrometer is used to measure parasites. The parasites are measured in order to identify what they are. Parasites that we look at can be a big range of sizes. Some of them are really small and some of them are really large. So to measure them will help you to identify it. And these have to be calibrated. I think it's annually. The micrometer has to be calibrated. Okay. This is another area of where these can be, the parasites can be found. You need to know these. In the sputum, Strongyloides, Paragonymous, Entamoeba, histolytica. If I just have like one name, like Strongyloides, it's the only one that we talk about, Strongyloides. But with Entamoeba, there are other Entamoebas, so that's why I have two names there. In the blood, Babesia, Plasmodium, and Trypanosoma. In the tissue, Trichinella, Leishmania, Pneumocystis, and Toxoplasma. These Trichinella is in the muscle tissue. Pneumocystis is in the lungs. Leishmania can be in the skin. In the spinal fluid is Naegleria and also Acanthamoeba. Okay, you're going to be studying what all of these look like, and you need to know how to go about identifying. So whenever you're looking at the specimen, you always look around on the 10x objective. If you see something suspicious, then you go to the 40, and first you decide, is this a troph or is this a cyst? With the trophs, like I said, they're elongated. You're going to look at what the cytoplasm looks like. You're going to look at what the nucleus looks like. In the nucleus, in the center, is something called a karyosome. Some of them have blot-like karyosomes. Some of them have real small, discrete karyosomes. Then the peripheral chromatin you want to look at. This is the chromatin around the edge of the nucleus. And you also want to look at the size, how big it is. Also, you need to look at how many nuclei the parasite can have. Some of them may have more than one. Okay, the cyst is more egg-shaped. It's transmitted through food or water. Excistation, that means that it transfers out of the cyst stage into a troph, is done in the lower intestine, and it transfers into a troph form, and then they start to multiply. They transform back into a cyst whenever conditions are not favorable, just like the spores are used in bacteria. You also look at the nuclei and how many it has, what the peripheral chromatin. You can see the peripheral chromatin is a lot thicker in this parasite compared, even though these are drawings, this one doesn't have any, and then these do. You look at the karyosome, the chromatid bodies or chromatid bars, it's the same thing. That's what this is. And then some may have a glycogen mass, which almost looks like just a big vacuole up here. Okay, laboratory ID. You find out if you're looking at a cyst or a troph, and then you look at the number of nuclei, the inclusions, what the cytoplasm looks like. Okay, this is the first of the parasites. This is Entamoeba histolytica. Out of all the Entamoebas, this is the only pathogen. The rest are not even treated. The trophs are 8 to 65, so they can be rather big, and that's usually due to their pseudopod. They have rapid motility and finger-like pseudopods, and that's what this is. They don't always have these. Like this, this is a troph, and it doesn't even look like it has a pseudopod, so they don't all have finger-likes. It has a single bullseye nucleus, which is referring to the karyosome. It has peripheral chromatin that's fine, even, and ground glass, and it can have red cells in the troph. This is a picture of the red cells. Now, since this is a trichrome stain, they will not stain like a red cell does on a right stain. So this is the organism nucleus and the karyosome, and all of these are red cells. This is the only organism that you're going to talk about, or you have to learn, that is able to engulf red cells. So if you see red cells, then you know it's histolytica. This is the cyst. It's 8 to 22. It can have 1 to 4 nuclei. So when you're looking at them under the microscope, sometimes you have to focus up and down a little bit to see all of the nuclei. You may be in like one plane and you can see this one, and then you move the fine adjustment knob and there may be one over here. So be careful. Focus up and down. The karyosome is small and central. It has fine, even chromatin in the periphery. The cytoplasm is fine and granular, and it has cigar-shaped chromatid bars. And that's this. They're supposed to be shaped like a cigar with rounded ends. And this one doesn't have a glycogen mass, but it can have a glycogen mass in it, like this one does. This is a direct stain. It doesn't have any iodine or anything in it. And this is the glycogen mass. It's just a storage compartment for glycogen. Okay, with histolytica disease, it can cause amebiasis. The trophs are in the intestines, and they can travel to the liver, brain, and lungs. It can also cause amebic dysentery. In the dysentery, the patients have a lot of stools. They can have flask-shaped ulcers in their colon. It's the leading cause of death by parasite. It's usually isolated where fertilizer, the farmers or whoever uses human waste, human stool as a fertilizer. And there's some different situations that this parasite can be seen, but it can really be anywhere. It can cause colitis. Colitis is infection of the colon in which the patient has diarrhea, they hurt in their stomach, weight loss, anorexia, fatigue, and flatulence. The dysentery, there may be blood in the stool. Extraintestinal amebiasis is when they can travel to other organs. They may form an abscess in the liver. The treatment is usually Flagyl or furamide. You don't need to know the treatments. You don't need to worry about memorizing the treatments. Okay, with histolytica, as you remember in heme, the eosinophils are what fights off parasites. Sometimes, I've never seen this, but you can see these crystals called Charcot-Leyden crystals, and these crystals are from the breakdown of eosinophils. This is a direct mount that's not stained, and then this is stained, and it's stained a pink color on a trichrome stain. But you need to know what those are, and those could really be in any parasite that we talk about, but it's seen with this organism more often. And again, this is the only pathogen. If you there's no pictures of parasites in your color atlas, but if you want to look at pictures in your book, Entamoeba histolytica is on page 648, and there are some very good comparison charts in that book, in your textbook also, that would be worth your while to study. Also, I want to mention that companies have came up with little ELISA tests that are immunoassays to look for some of these parasites now. The ELISA tests are similar to a pregnancy test, except of course you're going to use stool as the specimen. Hartmanni is the next one. This is Entamoeba hartmanni. This is not a pathogen. Of course, if it's there, it means the water is dirty or the food is dirty, but it's not considered the cause of the patient's diarrhea. The trophs are 5 to 15. You don't need to memorize the exact size of all of these, but I would pay attention to the size variation. What I mean is histolytica, the troph is 8 to 65, so these can get rather huge, but hartmanni only goes to 15, so these are not big. That's kind of what you need to, be able to tell is the size comparison. The motility is non-progressive, so it may be moving, but it's not going anywhere. It's not traveling to a different place. The nucleus, there's usually one. It's not seen on a unstained wet mount. It has fine peripheral chromatin in the nucleus with a beaded appearance. The cytoplasm is fine and granular, and it may have bacteria in it, but it does not have red cells. The cysts are 5 to 12. They can have up to 4 nuclei. It also has a small central karyosome and cigar-shaped chromatid bars with rounded ends. This is Entamoeba hartmanni troph. and this is the cyst. And you can see this little cyst, it looks like it's getting a little pseudopod right there, so maybe it's turning into a, oh, this is a troph, I'm sorry. No wonder it has a pseudopod right there. This is a Hartmanni cyst. This is a Hartmanni cyst. This is the nuclei, and these are the chromatid bars. I'm not sure what this is. It may be trash. Okay, E. coli. This is a one that's easy to identify also. This one is 18 to 27. It may be up to 50 micrometers. It has sluggish, non-progressive, blunt pseudopods. Instead of having finger-like pseudopods, you can't hardly even tell that it has one. It just will extend out a little bit. It has one nucleus, a large irregular caryosome. Let's see if we can tell. That one doesn't really look very. This one is a little larger. Vacuolated cytoplasm. The chromatin is coarse, granular, and refractile. This is around the nucleus. The cyst is 10 to 35. It can have up to 16 nuclei. So all the other ones in this group do not have a lot of nuclei. They cannot have any more than 4. So if you see a cyst and you count more than 4, then you know it's E. coli. 1, 2, 3, 4, 5, 6. So I know that this is Entamoeba coli because there's more than 4. And again, remember when you're looking at these in lab to focus up and down. You still may see another nucleus here that you can't see unless you focus it up and down. It has large, discrete, eccentric karyosomes, coarse granular chromatin. The cytoplasm on the outside is coarse. It has splinter-shaped chromatid bars that are pointed, and glycogen granules. And this one also is not considered a pathogen. This is a vacuole. This is some kind of inclusion, maybe yeast or something. These are all vacuoles, these little circles. I can't see any chromatid bars there. This is a huge vacuole, and I just included this picture because I thought it was odd. It almost looks like an alien face. But these are the nuclei, and then this is a large vacuole in the center. Okay, the next one is still in this group, but the first name is Indolamax. It's not Entamoeba. This one is 5 to 12, so it's rather small. Notice it has the name Nana. It's sluggish, non-progressive with blunt pseudopods, one nucleus. This one has a blot-like large karyosome, and you can tell that this one is a lot larger than the ones that we've, especially this one. This is rather big. It doesn't have any chromatin around the nucleus. The cytoplasm is vacuolated or granular, and it can have bacteria. The cyst is 4 to 12 micrometers, and it may be oval. It can have up to 4 nuclei, most commonly 4. A large blot-like karyosome, no chromatin, vacuoles, and a glycogen mass. These are the blunt pseudopods. Blot-like karyosome, blunt pseudopods, blot-like karyosome. This is the cyst. It looks like it's been eaten, but it's probably just broken off. These are the blot-like karyosomes. You can't even hardly see the periphery of the nucleus. Okay, Entamoeba bushleyi, this is also in that same group, except it doesn't have Entamoeba as the first name. Iotamoeba is 8 to 20 micrometers. It has sluggish, non-progressive motility, one nucleus, large refractive achromatic granules, coarse granular vacuolated chromatin, vacuoles in the cytoplasm, and bacteria and yeast as inclusions. The cyst is 5 to 22. It has one nuclei that's supposed to look like a basket of flowers, and I don't really see that, but if you notice I have it in quotations. This is not my description. It has a large eccentric karyosome, coarse granule and uneven distribution of the peripheral chromatin, a coarse cytoplasm, and where it gets its name from is the iodine, Iotamoeba. It stains really well with iodine, especially the glycogen mass that the cyst has. This is the cyst, and this is the glycogen mass. This looks like the nucleus over here maybe, and this is the big glycogen mass. This is the other glycogen mass. This is trichrome stain. This looks like a comparison chart, but it didn't come from your book. This comparison chart is not as good as the one in your book. You don't need to know this one right here with the little star. It's very rare, but you do need to know the other. So please look in your book at those comparison charts. They will help you. Okay, I like to talk about this one even though it's not a fun one. Naegleria fowleri. This is the organism that you've seen on the news that is referred to as the brain-eating amoeba. Just this year, now I got this information. I've heard about these cases the whole summer, but I went to look on the news to try to find how many exactly, and these are the only ones that I could find. So there could be more from this year. In June, an 18-year-old died. This one was real big in our news because it was from North Carolina in Charlotte. The teenager was from Ohio, but was in Charlotte, and this was in a river where she had went white water rafting. In July, somebody died from swimming in South Carolina. In July also, there were 3 that died after swimming in a river in Texas. In August, there's been a swimmer in Florida to die. And I think this last one, I think the news article said that the patient was in a private swimming, I forgot exactly what it called it. It didn't say it was a pool, but it was in a private pool, pond, I'm not sure exactly what. This is a free-living amoeba, meaning that it doesn't have to have a host to survive, so therefore it's in rivers and ponds and lakes and what have you. Also in this group of free-living amoeba are Hartmanella and Acanthamoeba. All of these like water places, rivers, lakes, air conditioning systems, humidifiers, dust, air, compost, vegetation. Okay. Naegleria, usually what happens is the patient is swimming and some of the water goes up their nose and the parasite migrates directly to the brain. It happens very, very fast. Within just a couple days, the patient could die. There are 3 stages: the amoeboid troph, which has pseudopods, the flagellate troph, which has flagella, and then the cyst. The amoeboid troph has slug-like blunt pseudopods, a large caryosome, and this is found in warm water, swimming pools, ponds, rivers, lakes. It can be anywhere. Usually, when they find out the patient has died from this, is when they do an autopsy and they find lesions in the brain. When they look at it under the microscope, the lesions have trophs, neutrophils, and eosinophils in them. There have been people to survive, and I'm not sure if I have this in the PowerPoint. Okay, according to the CDC, 3 survivors total. Okay, the flagellate troph is pear-shaped. That's supposed to be jerky movements, and it spins. It has 2 flagella, a large caryosome. The cyst is round with a thick double wall. It has a double wall. 1 nucleus, a large caryosome, and it's vacuolated. These are the flagellate trophs. This is a double wall cyst. Naegleria will eat bacteria. So this is a little test. I'm not sure how often this is done, but what you do is get E. coli growing on an agar plate, and you put a drop of the patient's spinal fluid on there, and if the parasite is there, it will eat the bacteria, and it will cause this tailing effect where it eats the bacteria away. It causes primary amebic meningoencephalitis. It happens really fast. There's neutrophils mainly in the spinal fluid, which is important because EOs are usually there with parasites. So the doctors may think that this patient has a bacterial meningitis. They have symptoms of meningitis, fever, headache, sore throat, stiff neck, and seizures. The treatment, this is an investigational drug which they attribute to saving these 3 survivors. I don't think that it's FDA approved yet, or maybe it just got FDA approved, but anyway, it's a new medication, and it's not used alone. There are other drugs that are supposed to be used with this, and but this is the drug that they think that has saved the patient's lives. And it says in your notes how to prevent the organism. If you have a swimming pool, then you can make sure that the chlorine in your pool is clean. But if it's somewhere like the river or the lake, you can't do anything about it. You can wear a nose clip, but other than that, don't go underwater. Try not to get water in your face or up your nose is the only thing you can do. Okay, Entamoeba. The trophs are 12 to 45 micrometers. They have sluggish motility with spine-like pseudopods. This is what their pseudopods look like. This is considered a pseudopod, the spines coming out the end. It has 1 nucleus, a large caryosome, vacuoles, and a granular cytoplasm. The cysts have a double cell wall. They're round with ragged edges, and it has a wrinkled look. Let me show you these. This is the little wrinkled look. This is a right stain. One nucleus, a large caryosome, and a granular cytoplasm. Okay, usually the identification is from brain tissue if the patient has encephalitis. This also can cause eye infections from people who wear dirty contact lenses. So we either can have brain tissue or corneal eye scrapings. The trichrome or Giemsa stain, which is very similar to the Reich stain, is used. There's also an indirect immunofluorescent antibody test, and this is the method of choice. They live in the soil and water. It's usually found in immunocompromised patients. Pseudomonas is known to inhibit Entamoeba. I don't know if it eats it or what exactly happens, but they don't live together. The disease, it can cause granulomatous amebic encephalitis. The patient has meningitis symptoms. It can cause lesions on the brain that causes personality disorders. There's no known treatment for the encephalitis infection. There is for the keratitis. This is usually seen from soft or extended wear contact lenses when the patient does not clean their contacts or clean their lens solution like they're supposed to. It causes ocular and vision problems, and neomycin drops or Broline can treat the eye infections. These are the spine-like pseudopods. This is the cyst with a double wall and wrinkled look. Okay, those are the amoebas. These are the flagellates. The flagellates have a flagella, of course, and these are some terms that you need to know what they are. The axoneme is an internal fibril that may turn into the membrane or flagella. In this axoneme, the axoneme you can hardly see. I don't even see anywhere on here, but it's a tiny fibril that, like a little dot, where the flagella may come off. The axostyle is a rod-like structure that's kind of like the spine of the organism. The costa is a rib-like body that supports the undulating membrane. So this black line is the costa. The cytostome is the mouth. This is this parasite's mouth. The median or parabasal body is a heavy fiber that supports the cytostome or undulating membrane, and it's not on there. It's not on here too. You just need to know what it is, and it looks like a little dot. The undulating membrane is this. This is a membrane that comes off the costa, and it's a fin-like membrane that helps the organism move. This is the undulating membrane. There's a membrane in here, even though it looks hollow. These are the flagella on the top, flagella on the bottom. This is the axostyle, and this is the organism's nucleus here. This is just a diagram, of course, but this is the undulating membrane. Okay, Giardia. This one used to be called Giardia lamblia, but they have changed the name to intestinalis. These are really easy to identify because they don't look like anything else. The troph is pear-shaped. It's 8 to 20 micrometers. It has a falling leaf motility, which this would be seen in a direct wet mount. They have 8 flagella or 4 pair. They are bilaterally symmetrical. So that means, if you don't remember, if you were to draw a line down the center of these parasites, it looks exactly the same on both sides. It's supposed to look like an old man, these nuclei being the eyes, and then I think the flagella are supposed to look like a beard maybe all these flagella coming off the end. It has 2 median bodies, 2 axonemes, and a sucking disc that's used to feed and to suck onto the patient's body. This is the cyst, and the cyst also is bilaterally symmetrical. They're oval, they look like a football. They have 2 or 4 nuclei. The immature cysts have 2 and the mature ones have 4. The cytoplasm is retracted from the cell wall. This is what this white space is around this organism. It has 2 or 4 median bodies and interior flagellar structures. These little lines are flagellar structures, and when it turns into the troph, they're going to extend out. This is a troph. You can't see the nuclei really well, but that's what that is. Here's the troph. These are trophs. This is a cyst. Giardia is known to cause really foul-smelling, explosive diarrhea. Of course, the stool is used. There are antibody tests that are immunoassays that are used now to quickly identify this. There's a test called an enterotest, in which a pill is used. The pill is attached to a string. The patient swallows the pill, and the string, the end of the string stays out of their mouth. The patient has to sit there and wait for the pill to be travel down into the digestive tract, and then they pull the pill back up by the string, and you look at the pill under the scope for trophs. They multiply by binary fission. They feed on the mucosa of the intestines. They can infect the bile duct and the gallbladder, and they also can live in water for up to 3 months. You need to know this. This is the most common cause of intestinal pathogenic protozoan in humans in the United States. Giardiasis is also known to cause a traveler's diarrhea. It can cause a malabsorption syndrome, especially in kids. It damages the intestinal mucosa, and the patient cannot absorb nutrients like it's supposed to. IgA deficient patients are more susceptible to getting this, and you need to know that. Flagyl is a treatment option for Giardia. Okay, chylomastics, meslei. The troph is 5 to 25. It's pear-shaped. It has stiff rotary directional motility. It has 1 nucleus with a small karyosome, a prominent cytostome, which extends 1/3 to 1/2 the length of the organism. It has a spiral groove that's said to give the organisms a shepherd's crook. The cyst is shaped like a lemon, and it has a clear hyaline knob on the top. 1 large karyosome, a well-defined nucleus. Iodine is used best to identify this. It makes the structures, makes you be able to see the structures better. Okay, so this is chylomastics. Can't really see anything on there. Okay, this is the nucleus up at the top. It has a typical spiral groove right here. The cyst is easier to identify. It has a knob on one end. This is the hyaline knob. It has a large karyosome. Okay, and also chylomastics is not considered a pathogen. Entamoeba fragilis, the troph is 5 to 18 micrometers. It can be irregularly shaped or round when it's moving. It has broad hyaline pseudopods. It can have 1 or 2 nuclei, but most commonly it has 2. The chromatin granules may be in masses of clumps or massed clumps of 4 to 8. It has vacuoles. This one does not have a cyst stage. You need to know that. It's thought to be transmitted by the eggs of Anaerobius, which is a pinworm, or Ascaris, because patients usually have both. So they may have Dientamoeba and Anaerobius, or they may have Dientamoeba and Ascaris. The infection is often asymptomatic, or it can cause a gastrointestinal illness. Okay, this is Dientamoeba, and this is most commonly 2 nuclei, and this looks like a vacuole. This is another one. I think it just has 1 nuclei. Okay, Trichomonas hominis. In urinalysis, and maybe micro too, I can't remember, we talked about Trichomonas vaginalis. Well, there's 2 pathogens in the Trichomonas family. Hominis is found in the stool. It's swallowed and it can travel through the intestines. So the specimen is stool. With vaginalis, this infects the vagina. So this is seen in urine or vaginal discharges. And they look a lot alike. The trophs are 7 to 20 micrometers. They're pear-shaped. It has jerky motility. The Trichomonas vaginalis also has jerky motility. It has 3 to 5 flagella, 3 to 5 anterior, 1 posterior. Can't really see them in that picture. It has a full-length undulating membrane. This is the undulating membrane, and this is all membrane in here. But notice it starts here and it ends here. So it has a membrane down the whole body. It has a cytosome and an axostyle. There is no cyst form. This is usually seen in children. It's asymptomatic. It's not even treated. It's usually thought to be in contaminated milk. Okay, vaginalis looks almost the same as hominis does. It's pear-shaped, jerky motility, 1 oval nuclei. All of the flagella originate anteriorly, and 3 to 4 extend posteriorly. They originate anteriorly, though. The undulating membrane is only half of the body, so it starts here and it stops here. With hominis, it goes all the way to the end. Okay, it has an axostyle. There is no cyst form. It's found in urine, vagina discharges, and prostatic secretions. The saline wet mount is the preferred ID. The best specimen is vaginal discharge, and you add saline and look at it under the microscope, and you can see the motility really easy. It usually infects women. They feed on bacteria in the vagina. It's transmitted by sexual contact. It's also been known to be transferred through wet toilet seats, underclothing that's wet, wet sponges, wet towels. It can live in water for up to 40 minutes. It can also migrate from mother to fetus. Trichomonas vaginalis, men are usually asymptomatic carriers. Women have a foul-smelling greenish-yellow vaginal discharge. They have burning and itching and red lesions. In infants, when it spreads from mother to baby, it can cause respiratory infections and conjunctivitis. it can be treated with Flagyl. Okay,